Dear students, welcome to the SDK Health Sciences Biochemistry classes. Our today's topic is the overview of metabolism. We will see the Gibbs free energy, enthalpy, entropy, carbohydrate and energy metabolism. Our aims and objectives for this lecture will be: we will define metabolism, we will classify the different types of the metabolism, we will compare the differences between the anabolism and catabolism, we will evaluate the Gibbs free energy, enthalpy and entropy. We will evaluate the energy metabolism in terms of endergonic and exergonic reactions, and we will see the energy yielding power of different food ingredients. We will evaluate the role of different organelles in metabolic processes, and we will recognize and interpret the pathways of carbohydrate and energy metabolism. And last but not the least, we will see the importance of carbohydrate metabolism. So metabolism is the sum of highly organized chemical reactions that takes place in an organism, or we can define it that metabolism is a phenomena that arises from interactions between molecules within the orderly environment of the cell. We can say that metabolism is comprised of a set of chemical reactions that modifies molecule A into molecule B, that modify molecule A into molecule B for what? Number 1, for energy. Number 2, for storage. Number 3, for use in some other reactions. And number 4, to make some byproducts. What are the processes that are included in the metabolism? They are cell growth, reproduction, a response to environment, survival mechanism, and sustenance and maintenance of cell structure and integrity. These are the processes that includes in totality the metabolism. If you see the type of metabolism, metabolism is a combination of two major processes. Number 1, anabolism, synthetic process. Another one is catabolism, the breakdown process. Now what happened in these two processes? If you see the larger molecule, we took in diet larger molecules. Say for example, we took starch, which is a bigger molecule. We took glycogen, which is a bigger molecule. We took protein, which is a bigger molecule. These larger molecules are broken down into glucose, monosaccharides, or amino acids in our body. So this phenomena in which the larger molecules are broken down into smaller molecules is called catabolism. When they are converted into smaller molecules, they release some energy as well. On the other hand, if a smaller molecule with the help of energy combine together, say for example amino acids, they combine together, or monosaccharides, they combine together, then they synthesize again the larger molecule in our body. This phenomena is called anabolism. So for example, amino acids, they combine to make protein, and monosaccharides or glucose combine to make a glycogen in the body. So larger molecule are broken down into smaller molecule, catabolism. Smaller molecule are combined to make a larger molecule, anabolism. Anabolism or anabolic pathways, they are the biosynthetic pathways. They consume energy, they use energy to build complicated molecules from simpler compounds, such as synthesis of protein from amino acid, as we discussed, synthesis of glycogen from glucose, and the energy coming from the catabolic pathways. So catabolic pathways gives energy. That energy is used by the smaller molecule to combine. The body use that energy to combine the smaller molecule and to make the larger molecule. This phenomenon is called anabolism. On the other hand, catabolism, it is described as the breakdown of complex organic substances into simpler compounds, with the release of energy, and this energy is captured in the form of ATP, this is a high energy compound, or some other high energy compounds. So catabolism is the larger molecules into smaller molecules plus energy, and this energy is captured as in ATP or some other high energy compounds. Say for example, carbohydrate, protein, fat, they are broken down in the presence of oxygen to carbon dioxide, water molecule, and release energy and hydrogens. These hydrogens, we can't let hydrogen free in our body, because when it will be free in our body, it will disturb the pH, and pH has a very narrow range. So whenever there is hydrogen has to be captured by some hydrogen acceptors, and when hydrogen is combined, I mean the oxidation and reduction process, so what will happen? The compounds are reduced, and these compounds are reduced equivalents or reducing equivalents, and then these reducing equivalents transport this hydrogen from, if it is in the cytoplasm, from cytoplasm to the mitochondria, or if it is in the mitochondria they will transport it into electron transport chain, and where they produce energy. Metabolism, anabolism, and catabolism. Anabolism is constructive biosynthetic, catabolism is destructive breakdown. Anabolism use energy, catabolism release energy. Anabolism is endergonic, catabolism is exergonic. Anabolism is like moving towards uphill, you are going up using energy. In anabolism, simple molecule forms the complex compound, such as carbon dioxide, water molecule. They combine with the use of energy to make glucose, amino acid, or lipid molecules. On the other hand, complex compounds are converted into simple molecules, such as glucose, amino acid, and lipids. They are broken down into carbon dioxide, water, and energy. Here the energy is released. Here the energy is used. Metabolism can also be classified in terms of normal and abnormal. Normal metabolism. Say for example, blood glucose. Normal fasting plasma glucose level is less than 100 mg per deciliter. Desirable low density lipoprotein cholesterol level is less than 100 mg per deciliter. HDL level, high density lipoprotein, desirable is more than 40 mg per deciliter in order to keep the body in a normal situation. Triglyceride desirable is less than 150 mg per deciliter. The normal BMI for an adult is 18.5 to 24.9. So this is normal metabolism. If this metabolism is disturbed, then it will turn to an abnormal metabolism. So say for example, abnormal metabolism example is if we are taking carbohydrate in terms diabetes. So if someone is pre-diabetic, his fasting plasma glucose level will be 100 to 125 mg per deciliter. And if the level is above, then fasting plasma glucose level is more than 126 mg per deciliter, then this person will be labeled as diabetic. LDL cholesterol, low density lipoprotein cholesterol, more than 100 mg per deciliter is abnormal. HDL cholesterol, high density lipoprotein cholesterol, less than 40 mg is abnormal. Similarly, triglyceride, more than 150 mg per deciliter is abnormal. And if we take the BMI of body mass index, so 25 to 29.9 is overweight or pre-obesity. 30 to 34.9 is class 1 obesity, and 35 to 39.9 is obesity, which is class 2 obesity, obese obesity. and more than 40 is extreme obesity or obesity class 3. So we classify the metabolism into anabolism and catabolism, and we classify the metabolism. Clinical classification of the metabolism is normal metabolism and abnormal metabolism. So in nutshell, what is metabolism? Metabolism move around energy and then maintain the life. So this is the trade between life, metabolism and energy. So metabolism related to energy and energy related to function of the life. So this is the triad of metabolism, energy and life. The chemistry of life is organized into metabolic pathways. In human body, the chemistry is organized in terms of metabolic pathways. Organism transforms energy and live at the expense of energy. They transform energy from one form to another form and then they live at the expense of energy. ATP is the currency of energy that provides cellular power to the cellular work by combining or coupling exergonic reaction with the endergonic reactions. Or we can say they combine energy yielding, energy releasing reactions with energy utilizing reactions. Some of the metabolic processes release energy, other metabolic processes use energy. What is the major role of the metabolism? The major role of the metabolism is to capture chemical energy from foodstuff as ATP and utilize that ATP for cellular functions. The respiratory function, cardiovascular function, neurological functions, cell transport, ionic transport, transport across the cell membrane. All these are the functions of the muscular functions. All these are the functions of the body. Human can generate ATP by oxidizing carbohydrate, fatty acids and amino acids. The quick source of energy is carbohydrates. Large source of energy is fatty acids. And amino acids are used at the last resort. At the simplest level, energy homeostasis involves balance between the fuel intake and energy expenditure. There should be a balance between intake and expenditure. If this balance is disturbed, then what will happen? If intake is more and expenditure is less, obesity. If intake is less and expenditure is more, then starvation. So we need to keep the balance between the intake and the expenditure. Now, how to define energy? Energy is the capacity of work, to do work. Capacity to do work. Now, what we do, what the human body is doing, that it taking chemical energy from the food stuff, which is measured in kilocalories, sometimes denotes as calories, and then transfer this energy, which is taken from the food, into energy currency in the body, that is ATP, and then utilize it for its different functions. Now, if you define the energy, we say 1 kilocalorie is defined as the energy required to raise the temperature of 1 kilogram of water by 1 degree Celsius. So this is 1 kilocalorie. 1 kilocalorie. Now, what happened in the body? Biomolecules from outside, say for example carbohydrate, fat and protein, they are catabolized, oxidative reactions are there, exergonic reactions are there, energy is released, and then they are converted into carbon dioxide, water, and if it's coming from amino acid protein, then ammonia. Then what happened in the middle? They release energy in the form of ATP. They release NADPH. These are required for the synthetic processes. So smaller molecule, amino acid, sugar, protein, nitrogenous bases, they combine together in the reductive mechanism, endergonic reactions, where they need energy, anabolic process, and then they synthesize protein, polysaccharide like glycogen, lipids, nucleic acid in our body. In the middle of this, what happened? There is one more thing which is released, that is called hydrogen. Hydrogen is released. As I said earlier, hydrogen is, we can't allow hydrogen to be free in circulation in the body. So what will happen? There are hydrogen acceptor, NAD and FAD. They are the hydrogen acceptor. They accept the hydrogen. They are oxidized. When they accept the hydrogen, they become reduced. And these are called reducing equivalents. These reducing equivalents enter into the electron transport chain, and in electron transport chain, they give us energy. If they are coming from cytoplasm. If they are already, the mechanism is already present in the mitochondria, then from the mitochondrial, from the site of the reaction, they will enter into the electron transport chain. So, this is mitochondria, this is cytoplasm. Let me make mitochondria like this. This is nucleus, this is cytoplasm. So, if NADH is in the cytoplasm and this in the mitochondria, there is electron transport chain. So, they will go here. And if it is already present in here, so they will enter into the electron transport chain. The transport of NADH, the hydrogen of NADH and FADH from the cytoplasm to the mitochondria use shuttle pathways. So they can't go directly, but rather they are going through the shuttle pathways. What happened here? Metabolism of the carbohydrate, fat and protein. This is a subset with the hydrogen. The hydrogen will be released. When the hydrogen is released, there is a hydrogen acceptor. These hydrogen will be accepted and then they will be converted into NADH. And this carbohydrate, fat and protein, they will be converted into oxidized substrate with the carbon dioxide with the water molecule. Now this NADH, it will enter into the, it will combine with the water molecule and the electron transport chain, release the hydrogen, convert it into oxidized form of the NAD, and in the process of oxidative phosphorylation, what will happen? There is an oxidation. It will take the hydrogen, convert it into water molecule, and with the transfer of electrons, energy is released, and energy is this much. The amount of energy will be so much that it will adhere in organic phosphate with the ADP to make ATP. And this ATP will be utilized in the muscle contraction active transports. When the ATP is utilized, again it will phosphate will be utilized here, and then it will convert into ADP, and the cycle will continue. The cycle will continue. The energy yielding capacity of the different metabolic fuels. So say for example, if we see 1 gram of the carbohydrate, it gives us 4 kilocalories. If we take 1 gram of the fat, it will give us 9 kilocalories. And if we take proteins, it will give us 4 kilocalories. These are the energy yielding or energy releasing capacity, heat of combustion capacity of the fuels, energy fuels in our body. So we consider these bioenergetics. Let's see these bioenergetics in terms of law of thermodynamics. So we have first law of thermodynamics and we have second law of thermodynamics. What is first law of thermodynamics? First law of thermodynamics says that energy can neither be created nor destroyed. So it can neither be created nor destroyed. So what does it mean? It means that the energy generated in any system is energy that has been transformed from one state to another state. So it can be transferred from one form to another form, but it cannot be generated nor be destroyed. Similarly, the second law of thermodynamics, it said that efficiency of energy transformation is never equal to 100%. Is never equal to 100%. So, first law of thermodynamics, energy cannot be created nor be destroyed, but it can be changed from one form to another form. Point number 2, the second law of thermodynamics, they said that efficiency of energy transformation, if you transform from one form to another form, it should not be 100. It cannot be 100%. It cannot be 100%. So if it cannot be 100%, then what happened? It means that something is going in between. Something will be lost. So therefore, all processes lose energy. And they lost energy as heat. Conversion to the heat is the ultimate fate of the chemical energy. So what happened here? See, for example of the first law of thermodynamics. It is taken from this site. So first law of thermodynamics, this cheetah is eating this deer, and it taking energy from this flesh, from this stuff of the deer. So first law of thermodynamics cannot be transformed, it cannot generate, but it is taking food stuff from here, and then that is converting into another form of energy. So this chemical which is eating, this will convert this chemical energy into kinetic energy for movement, for running, for all these activities. Energy can be transformed, neither created nor destroyed. For example, chemical energy, the potential energy in the food will be converted into kinetic energy in the movement. So this is the simple. So food, we take food, and the chemical energy here we transform into kinetic energy that we use for our movement or heart function. The second law of thermodynamics, what it says? The second law of thermodynamics says that when you transform the energy, the outcome is not 100%. So, we take food energy from here. This food energy is converted into kinetic energy. The outcome is carbon dioxide and water. But in between here, there is a loss of energy. There is a loss of energy. So, say for example, 100% energy from here. At the end of the day, we could have 80%, 20% is lost, 20% is not, and this lost is especially in the form of heat. We say that biological system, the biological order and disorders, how the biological system it works. We know that biological system is comprised of, in the living system, it cannot be static. It cannot be static. If it is static, there is no life. For the life processes, there must be processes of anabolism and catabolism. Two processes are going side by side. Some of the things are synthesized, some of the things are broken down. So living system increases the entropy in the universe. It uses energy to maintain order, keep things in normal situation. And we use Gibbs energy equation to see the normal outcome. Which type of the reaction is this? To see the type of the reaction. So delta G is actually the change in the free energy of a system that is moving from initial state of substrate to the final state of product. How we can classify the delta G or change in the energy? Delta G is equal to delta H, which is enthalpy minus time into delta S, which is entropy. So change in energy is equal to enthalpy minus times into entropy. Now what are these entropies and enthalpies? Enthalpy is the amount of the heat that evolve or absorb in a reaction, or the amount of the heat that the molecule have. Whereas entropy is a disorder, as we discussed earlier. It is the disorder during the transformation of one form of energy to the another form of energy. And second law of thermodynamics says that it could not be 100%. That's why some free heat is released, and that heat is created disorder in the universe, and that is known as entropy. Based on this entropy and enthalpy, the delta S and delta H, we can say that a reaction could be an endergonic reaction or exergonic reaction. A reaction could be a reaction that needs energy or that releases energy. Two kinds of the reactions are there. So number one, if the free energy of a reaction tell us whether the reaction is endergonic. If it is endergonic, delta G will be more than 0. Or if it is exergonic, spontaneous reaction that happened without adding energy, that will be exergonic reaction, or delta G will be less than 0. Delta G will be less than 0. It cannot be 0 at any time. 0 mana input and output is equal and then the person is dead. So in living system, it should be either exergonic or endergonic. So less than 0, a negative delta G means that the reactant, substrate or initial state have more free energy than the product or the final state. They are called exergonic reactions, and they are also called spontaneous reactions because they can occur without the use of extra energy. So A and B, if they have powerful energy system, the initial state have more free energy, then they can start their reaction and they can convert into product. If the power of energy is more in the substrate than the product. Delta G more than 0, mana it is these reactions are the endergonic type of the reaction. The positive delta G requires an input of energy. In order to complete the reaction, the reactant needs energy, and they are called endergonic reaction. And in this case, the product or the final state will be having more energy as compared to the reactant or initial state. Endergonic reactions are non-spontaneous because they need energy, meaning that the energy must be added before they can proceed further. So we can see the example of endergonic reaction and exergonic reaction. Endergonic reaction, delta G is more than 0. So say for example here, you can see here this is a nitrogenous base. Say for example adenine. This is nitrogenous base. This is ribose sugar, 5 carbon sugar, and 2 phosphate are attached to this. 2 phosphate are attached to this. This is ADP. In ADP, the outer phosphate has the capacity of 7.3 kilocalorie. The power of this, the energy, this phosphate bond is having 7.3 kilocalorie. When it will combine with another inorganic phosphate, it is also having the capacity of 7.3 kilocalorie. Now individually, these two have less energy as compared to the product. The product have 2 phosphate together. One product have 2 phosphate together. And one is 7.3, another is 7.3, in total of 14.6 kilocalorie. So the substrate has less energy than the product. When the substrate has less energy than the product, so the product, the final state, have more free energy as compared to the reactant or initial state. So this reaction is endergonic reaction. This reaction is endergonic reaction. This is non-spontaneous reaction, and these reaction needs extra energy to start to convert into product. And this is endergonic reaction. See, nitrogenous base, phosphate, inorganic phosphate, and ATP 7.3 kilocalorie. Say for example, the reaction needs energy. Say for example, it's coming from the sugar. It needs energy, and final product is the endergonic reaction. These are the reactants. If we draw a graph, free energy, the Gibbs free energy delta G into times. The reactant starts here. They have the free energy to some extent, but they cannot move forward unless the extra energy is added to them. So extra energy has been added. They move up. They reach to a state where they make the product. This energy, which is the extra energy, is delta G, and which is more than 0. This is endergonic reaction. So endergonic reactions, the reactant individually, the reactant or the substrate has less energy as compared to the product. They need added energy. And then the delta G is more than 0. As compared to this exergonic reaction, the delta G is less than 0. And this type of the reaction where the reactant or initial state have more free energy as compared to the final state. So reversal of this reaction. Say for example, now what we are doing, we are breaking the ATP into ADP and inorganic phosphate. So ATP has higher energy. Water molecule is added, hydrolysis has been done, and then hydrolysis of the ATP will convert ATP into ADP and inorganic phosphate, and individually they have 7.3 and 7.3 kilocalorie. So what happens? Substrate has high energy as compared to the product. Delta G is less than 0 because and reaction is spontaneous without added reaction. So you can see here 7.3 kilocalorie is released. When the amount is released, so it will be designated as negative sign. So minus 7.3 kilocalorie. ATP. What is ATP? ATP is the cell energy, adenosine triphosphate, and it is used for a cellular function. So body utilize ATP for cellular functions. It's a combination of nitrogenous base, ribose sugar, and 3 phosphate. 1, 2, and 3. Phosphate is in the form of H3PO4. So this is ATP. Energy is released from the ATP. The terminal phosphate is broken down. So this terminal phosphate is broken down, and when this bond is released, this bond has energy of 7.3 kilocalorie. If you want to add this bond again, you need a reaction that will release more than 7.3 kilocalorie. 7.3 or more than 7.3 kilocalorie, then it will be able to add this phosphate to this ADP, the phosphorylation. So reversal of this. Now we can analyze this by some examples. Say for example, this is a glutamic acid, which is an amino acid, combined with ammonia, it forms glutamine. The total reaction is delta G is 3.4 kilocalorie plus. So this is an endergonic reaction. Endergonic reaction, delta G is positive. It is not spontaneous. Mana, when glutamic acid was combining with the ammonia, it needs added energy. There should be energy, added energy. We know that in the body, individual reaction do not take place. Definitely, there is a combination. There is a combination. Anabolism and catabolism going side by side. So what happened? This is reaction number 1 where the delta G is positive. What happened in this reaction? Simultaneously, as we said that added energy is required. So what will happen? ATP will be utilized. And when ATP is utilized, so with the help of the water molecule, ADP will form an inorganic phosphate is released. Because energy is released, this is minus 7.3 kilocalorie. This reaction is exergonic. Now if we just sum all these three, so we have delta G positive, we have delta G negative 7.3 kilocalorie, and then in the coupled reaction combined, we can see that it is minus 3.9 kilocalorie. Delta G is minus. So this reaction of glutamic acid, combination of ammonia to make glutamine with the utilization of an ATP to make ADP with the removal of inorganic phosphate. This coupled reaction, overall this coupled reaction is a spontaneous reaction. A spontaneous reaction. That is how we analyze the reactions in our body, and our body took place such a reaction in such way. What is the metabolic role of organelles in our body? We have cytoplasm, nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, and lysosomes. In cytoplasm, carbohydrate metabolism took place, protein metabolism took place, fat metabolism took place, and nucleic acid metabolism. For carbohydrate metabolism, we have glycolysis, we have glycogen metabolism, we have HMP shift. For protein metabolism, protein synthesis, transamination, heme synthesis partially, urea synthesis partially. Fat metabolism, fatty acid synthesis, cholesterol synthesis. Nucleic acid metabolism, purine pyrimidine synthesis. These areas have enzymes, required enzymes for all these reactions. Similarly, nucleus, DNA replication. Similarly, mitochondria, the metabolic hub, have the enzymes for all these reactions. Electron transport chain, ATP generation, tricarboxylic acid cycle, beta oxidation, ketone bodies production, then urea synthesis partially, heme synthesis partially, gluconeogenesis partially, and pyrimidine synthesis. Similarly, endoplasmic reticulum have the enzymes for biosynthesis of the protein, glycoproteins, lipoproteins, drug metabolism, and synthesis of the cholesterol. The Golgi apparatus or Golgi body processing the synthesized protein, such as modifying, packaging, transportation of the protein of lipid to the site of requirements. Lysosomes have lysosomal enzymes which are required for the degradation of protein, carbohydrate, lipid, and nucleotides. These are the metabolic role of the organelles in our body. Dear students, now we will continue with the carbohydrates metabolism, the introductory components of the carbohydrate metabolism, and we will start with the definition of the carbohydrates. We'll say what are carbohydrates. So as you know, I hope from previous understanding, you know that carbohydrates are the polyhydroxy alcohol containing aldehyde or a ketone group. Or we can say that they are the compound of carbon, hydrogen, and oxygen. Where hydrogen and oxygen, they are present in the same proportion as it is present in water. Generally, they are derived from the plants, but our body can also synthesize them from other compounds. So, the simplest structure, the simplest one of the carbohydrates are the monosaccharides. So, for example, 6-carbon compound, glucose, galactose, fructose. Glucose and galactose are aldohexoses, and fructose is a ketohexose. Once we take carbohydrate in diet in a complex form, in the form of starch. So we took carbohydrate in the form of starch, broken down into the body, in the stomach, then in the intestine, and then from here it enter into the bloodstream. So they enter into the bloodstream in the form of monosaccharides. So galactose, fructose, glucose, and then they are absorbed to the blood circulation, and from here they goes to the our organs. So liver. Then liver, you can see that liver take all these three, but they release them in the form of glucose. Then it goes to the muscle, utilized there. It goes to the brain, utilized there. It goes to the adipose tissue and utilized there. We have different pathways for carbohydrate. Some of the major pathways that are involved in the carbohydrate metabolism, they are glycolysis, breakdown of glucose, gluconeogenesis, synthesis of the new glucose, glycogen metabolism, glycogenesis, and glycogenolysis. And then we have alternative pathway, that is pentose phosphate pathway, which is also called HMP shunt. We have some of the major pathways that are involved in the fuel metabolism. Glycolysis, or we can say energy metabolism. Glycolysis, breakdown of glucose gives energy. Citric acid cycle gives energy, and oxidative phosphorylation provides energy. What is the role of carbohydrate in our body? As we said earlier, it is the primary role of the carbohydrate as a fuel source for basic life function and exercise by providing energy in the form of ATP. They provide ATP, adenosine triphosphate, energy currency. How ATP is generated? ATP is generated by the process of phosphorylation. What are the types of the phosphorylation? We have two types of the phosphorylation: oxidative phosphorylation and substrate level phosphorylation. What is oxidative phosphorylation? Oxidative phosphorylation is also called chemoosmosis, or a process in which reducing equivalents take hydrogen from the site of the reaction to the mitochondria into the electron transport chain, where the reaction took place with the help of several enzymes, and it provides a large number of energy, or huge amount of energy. Substrate level phosphorylation, it is the type of the phosphorylation where we get energy at the site of reaction. So this is the major role of carbohydrate in our body. Provides quick energy. 1 gram of carbohydrates gives us 4 kilocalories. And it gives 4 kilocalories with the help of these two phosphorylation mechanisms: substrate level phosphorylation and oxidative phosphorylation. I hope everything is clear now. We will finish here by summarizing the whole session. Metabolism is the sum of all processes that allow cell or organism to extract energy from the environment and to synthesize macromolecules that are required for the life. Metabolism can be divided into two: anabolism and catabolism, or clinically it can be divided to normal and abnormal. Chemistry of life is organized into metabolic pathways. There are different metabolic pathways. Organisms transform energies. The energy transformation of the life is about subject to the two laws of thermodynamics. First law of thermodynamics: energy cannot be created nor be destroyed. Second law of thermodynamics: transformation is not 100%. The reaction may be exergonic thermodynamically, delta G is less than 0, or endergonic, delta G is more than 0. ATP is generated, which is the currency energy, and it is obtained by two mechanisms, that is substrate level phosphorylation and oxidative phosphorylation. ATP powers cellular work by coupling exergonic and endergonic reactions. Glycolysis, gluconeogenesis, glycogen metabolism, pentose phosphate pathway are the main pathways for the carbohydrate metabolism. Major pathways that involve the fuel metabolism or energy metabolism of the carbohydrate are glycolysis, citric acid cycle, and oxidative phosphorylation. Abnormal carbohydrate metabolism lead to diabetes or glycogen storage disease. Thank you very much, students. If you have any questions or any comment, please let me know. Your comments and feedback will be very important for me to modify to improve these sessions. Thank you very much. Best of luck.